All right, good morning. As I promised you yesterday during the course that we were going to start with molecules and build up to cells and tissues and organs. And so the first molecule, when you talk about living things, is DNA. And so what we're going to do today is we're going to talk about aspects of DNA replication that have particular importance in medicine and pharmacology particularly. There's several of the drugs that are used in the clinic inhibit DNA replication, and we're going to show some examples of that today. So this summary of learning objectives kind of gives you the big picture of what we're going to do, at least in the first part of the lecture. The second lecture today is going to be on genetic recombination. So once we learn how the DNA is copied, how it's made, we're going to learn a little bit about how the sequence can be reorganized to generate genetic diversity on the positive side and to contribute to disease on the negative side. Now, the way I've structured all this is we're actually going to do this in three parts because the replication story is pretty long and kind of complicated. So I'm going to try to do about 25 minutes. We'll take a few minutes, 5-minute break. We'll do another 25 minutes or so, take a break, and then we'll do the recombination story. Okay? Now, if I forget to turn the integrity thing on and off, somebody wave at me. But actually, I have the screen on today. I got the integrity on today. Banner day. All right. So let's begin the discussion thinking about some terminology that you probably already know, and that is concepts of chromatin and chromosome structure. We're really going to start with the clinical case that we introduced yesterday. Now, this is the PL, is the 16-year-old boy diagnosed with the Philadelphia positive acute lymphoblastic leukemia. And if you remember from yesterday, he was treated with some chemotherapy agents. And then after several weeks of therapy, the physicians would do some blood tests, basically to see if the chemotherapy was working. And those blood tests are complete blood count and differential count. And then this is what happened. So let's look at this for a second. White blood cells, platelets, lymphocytes, neutrophils. These are all normal constituents of blood, right? Lymphoblasts are actually not normal. They're not supposed to be there at all. And if you look at the normal spans or normal ranges, you can see that some things are out of whack. Got way too many lymphocytes, not enough neutrophils, not enough platelets, and there's lymphoblasts in the blood. And when a bone marrow biopsy was taken, 85% or 95% of those cells are lymphoblasts, and that too is wildly incorrect. Okay? So this is an example of how you use data. You use information that the patient presents, signs, okay, and symptoms that they report, and you try to sort out what's going on and what to do. And in this case, the physicians have treated with this cancer chemotherapy regimen, and it's not really working very well. So what can be done? Well, a couple of things can be done, and in this pretend scenario, adriamycin, a drug which is used in a variety of cancers, is going to be used. Now, this drug goes by the name of doxorubicin. Now, you'll see, just to introduce a common approach that I use for drugs, the little R means this is a trademark. So adriamycin is a brand name of the chemical doxorubicin. Us pharmacologists always use the chemical name whenever we can because there'll be several companies that may make the same drug. In the clinic, the physicians and nurses almost always use the brand names. So you just have to understand that. On the exams, my exams, board exams, the chemical names will be used. Brand names will not be used. But I'm giving you this just because there are some drugs like adriamycin, everybody on earth calls it adriamycin. It's just the way it works. All right? So anyway, the story here is this drug's going to be added to the regimen to try to get a better therapeutic response. And then the question is, well, how does this drug actually work? And the short answer is it inhibits DNA replication. And we're going to show you exactly how it works a little bit later in the sequence. Now, before we get there, let's stop and talk a little bit about this patient's Philadelphia chromosome. So what actually happened in this individual? Well, at some point, what happened was there was a chromosomal translocation. A piece of one chromosome broke off. In this case, chromosome 9 broke off and recombined with a piece of chromosome 22. And this is a very famous type of translocation that results in a very famous structure called the Philadelphia chromosome, this little stubby chromosome. And what you see here is BCR-ABL. BCR means breakpoint control region. That's not an important factoid, but that's what it means. ABL refers to a tyrosine kinase called the Abelson tyrosine kinase. That is an important factoid to remember. The result of this translocation is that there's a fusion protein that's created, and the tyrosine kinase is active constitutively, meaning it's on all the time. You say, well, why would that matter? Why does that make cancer? It creates cancer conditions in this individual and in others who have other types of cancers because that kinase controls the cell cycle. So DNA replication continues, cells continue to grow, and if this translocation occurred in lymphoblasts, you get lymphoblastic leukemia. If it occurs in myeloid cells, the patient would suffer from chronic myelogenous leukemia. And there are other leukemias as well that have, and other disorders that have Philadelphia chromosomes. Okay? You see what's happening? So our goal today in the next couple of days is to figure out the basic structure of chromosomes, how they're replicated, why this translocation might happen, how could it be repaired. Okay. So let's look at the fundamental structure of chromosomes to remind you that there are specialized regions within chromosomes that are important to understand. The centromere, shown here, is an area of heterochromatin that's really important in chromosomal separation during mitosis. For DNA replication, a really important region that we want to understand is the origins of replication. Okay, molecular geeks would call this an ORI, or an ORI, an origin of replication. Another important area we'll meet at the very end of the lecture, and it's at the end of the chromosome here, is a structure called the telomere. That's a piece of heterochromatin that's really important for the stability of the chromosome structure, and therefore the genomic stability of the individual and eventually the lifespan of the cell and the lifespan of the individual. So we'll learn more about that in just a second. All right, so replication is what we're going to talk about today. So this cartoon is meant to show you that the human chromosomes have DNA that are a long, linear, double-stranded, anti-parallel molecule that have specific sites where replication occurs, and the replication will run in both directions, and there are multiple sites along the chromosome. The result is you're going to get a duplication of the chromosomes, and of course, once the chromosomes become highly condensed and wrapped up, then during mitosis they get separated into the daughter cells. And you all know this from basic biology. So what's new for today? Probably what's new is we're going to need to think about a little more detail of how that chromosome is actually assembled. How do you get the human genome, 3 billion bases in the haploid genome, 6 billion in a diploid genome, how do you get that DNA packaged into a little itty-bitty space of the nucleus? One calculation is if you take all the genomic DNA and stretch it out, it would be 6 feet long. So how do you get that into the nucleus of a cell? And the answer is there's a lot of superstructure on top of just the fundamental DNA. Now, everybody's studied DNA, and you know it's a double helix, right? And I ask you to do some reading to remind yourself about how bases bind, how they stack, how they base pair, and so forth. So you know all that. In addition to that fundamental chemistry of the helix, most of the DNA in the cell is actually never in that sort of naked Watson-Crick model of DNA. Okay? Most of the DNA, most of the time, is wrapped up with proteins. And this introduces the concept of a nucleosome. So the arrow is pointing at these little yellow barrel, which is an octamer of a protein called histones. Okay? Important histones. And this, about 200 base pairs shown in red, wrapped around the histone, makes up a nucleosome. Now it's even more complex. The nucleosomes themselves get folded up, basically twisted up into a larger fiber referred to as a 30 nanometer fiber. The number's not really important, but it's just the concept is it gets folded up even more. Those fibers themselves get rolled up and folded up and twisted up even more into large loops of DNA, of chromatin, actually. And these loops get further folded up into supercoils, which get further rolled up into more supercoils bound to a whole variety of proteins to actually form the chromosome. So the chromosome is not just DNA. It's a whole bunch of proteins doing a lot of interesting things, one of which is replication of the DNA itself. So it's going to be important to have the concept of this structural complexity, and probably the most important idea presented here is the nucleosome. What is the nucleosome? So you want to know that and write that down if you don't know what that means. Okay. So one convention that we should introduce here that's going to be real important for all your courses in the future, particularly pathology, hematology, and oncology, cancer chemotherapy, and a variety of pathological conditions that are due to genetic diseases. And this naming terminology has to do with the structure of chromosomes and how genes are addressed, how their addresses are assigned. And they're based on this fundamental structure that the chromosome has two arms. This would be the chromosome during the M phase, right? All wrapped up and bound up. There's a short arm called the P, a long arm called the Q, and there are addresses along these two arms that are coded by numbers. So the ABL gene coding for the Abelson tyrosine kinase, which is the guy that's gone off the reservation here and the patient with AML. This gene is located on chromosome 9 on the Q arm, position 34.12. So this number refers to the position along the chromosome. And so in the human genome, all the genes have been sequenced, the intergenic regions have been sequenced, and so there's addresses known for the human genes. So you need to be familiar with this terminology. You need to recognize when we're talking about gene X is on chromosome 11 at Q13.7, that's the specific address of that gene. You say, well, oh, that's cool, but so what? Why would I use this? Well, pathologists use it to keep track of where genes are. Geneticists use it to keep track of which genes cause which traits and which diseases. And so it's a really important addressing scheme that you just have to know about. Now, bless you, do you have to remember all this stuff for the test? No. But the point is, this is how genes, in this case that cause AML, are described and how they're addressed. Okay? You get it? All right. The link at the bottom, FYI, is a link to the online Mendelian Inheritance in Man database. So a very famous geneticist at Hopkins years ago started collecting which genes cause what kinds of diseases. And as the molecular information became more and more available, then this database now has a collection of entries just like this for thousands of genes that are known to cause disease. And that's how you would find where is the gene that causes the disease that I'm interested in. So you can look that stuff up. All right. So, replication. We put the chromosome together. We know a little bit about the kinds of structures. We know that there are genes that go bad. They have addresses. Fine. But what about DNA replication in terms of medical practice? Why do we need to know about details of DNA replication? A couple of reasons. normal development clearly relies on DNA replication. So your patients, the growth of their children and so forth, critically reliant on DNA replication. Bacteria that will invade your patients and cause diseases, viruses that invade your patients that cause diseases, and the drugs that you use to treat the viruses and the bacteria causing these diseases, many of which act on DNA replication. So we got to know something about this process. And so the way we're going to approach this is we're going to sort of compare and contrast prokaryotic and eukaryotic. A couple years ago, one of the students said, well, why do we have to know about prokaryotic? Well, because bacteria cause diseases, that's why. And so in all cases of both prokaryotes and eukaryotes, DNA replication is considered semi-conservative. What does that mean? It's sort of half conservative, right? Does that mean it's partly liberal, or what's going on? That's like a little joke. Okay. What it means is that here, the DNA, you can see the two strands, both coded orange. So each strand serves as a template, right? And so the new strand in red is synthesized based on the template. And in this first round of replication, there's still two orange strands. In other words, this DNA molecule has been conserved, right? But only one of the two strands, half, semi-conservative. So that's what that means. And so this goes on in multiple rounds of replication, but you can see down here, by 3 rounds of replication, you still have the original orange strands in at least 2 out of the 8 DNA molecules. Okay, so everybody, I think, pretty much appreciates that. So how does this all happen, though, biochemically? And it differs in prokaryotes and eukaryotes. Now, what happens in prokaryotes, which in most bacteria, the DNA is organized in a double-stranded DNA in a circular genome. And that's what's represented in this cartoon. Okay, not all, but most bacteria have a genome that looks like this. Viruses are different. They're generally linear genomes. They're weird things. But in all cases, there's an origin of replication where the DNA synthesis starts. Key feature of the origins of replication is they're very rich in AT base pairs. All right, let's think back to basic energetics of base pairing. Why are AT base pairs interesting and important at the origin of replication? Yeah, because how many hydrogen bonds in an AT base pair? How many in a GC base pair? Got it. Okay. So ORIs are frequently AT rich. Now, that doesn't mean every AT rich is an origin, right? And exactly what the origins are, where they are, is an active area of research. Okay? But the point is, in bacteria, there's generally 1 per circular piece of DNA, and these replication forks form, and then DNA replication occurs in both directions and eventually duplicates the genome. Now, in eukaryotes, in us, it's more complicated. E. coli, 4.6 million bases. People, you got 9 billion in the diploid genome. All right? And they're separated onto different chromosomes. So it's a lot more complicated. And if you just do the math, the E. coli can replicate maybe 20 minutes under ideal growth conditions, and that's 4 million bases. How do you get 9 billion bases in sort of a reasonable time frame, an hour or 2? And the answer is you got multiple origins of replication throughout the various chromosomes, and that's represented here in the cartoon. And again, same principle, AT-rich regions, easy to melt, easy to get them apart, easier than GC-rich regions. Bidirectional replication going in both directions. And eventually you get nicely copied strands. So let's look at this in a little more detail. So you got an AT-rich region, and it can melt more easily, but still the preferred configuration is for the ATs to remain bound. Yes? So how do you get them apart? You need energy, and particularly you need a family of enzymes called helicases. The structure is a helix, so a helicase is an enzyme that pulls apart a helix. All right? So the DNA can be pulled apart by this helicase, which uses a little bit of energy to do that. Now you've pulled apart the DNA strands, okay, so they're out flapping around. So what happens if you have antiparallel DNA strands that have complementary sequences left to their own devices? It's kind of like teenage kids in the basement, right? There you go. Now, in order to prevent that from happening, you've got chaperones, right? Single-strand binding proteins in this case, the little orange circles. What they do is they bind the strands and keep them apart so that what can form is a very complicated group of proteins called the replisome, the place where DNA is replicated. So you start to unwind them with helicases, bind it all up, stabilize things with the binding proteins. Now, there are analogous proteins in both humans and bacteria. They're not identical, but they're functionally quite homologous. So what happens next? You've got to solve a couple problems. One is, if you're pulling apart the helix, this is attached to the rest of the genome. And you can imagine taking a rope and pulling the strands apart and start pulling it. What happens to the rest of the rope? Well, it spins around. So if the rope's tied to something like your boat or the dock, what happens is, as you pull apart the strands, the rope gets coiled. And it's eventually going to get tangled, and that's going to stop you from pulling it apart. Now, the solution to this in the cell for DNA replication is two types of enzymes called topoisomerases. Okay? An isomerase is an enzyme that changes the configuration or structure of a molecule. The topoisomerase changes the structure of DNA. And in this slide, what this represents is an example of how a topoisomerase can solve this problem of supercoiling. Let's go to the next slide. So this is a type 1 topoisomerase, and I want you to know the difference between type 1 and type 2. It's easy to remember. Type 1 makes one cut in the DNA strand, shown here as a nick. Right? Here's a hole. So you can imagine if you cut one strand, what that would allow the molecule to do is to rotate around the other strand. And that's exactly what happens. And so the type 1 topoisomerase can kind of prevent new coils from being introduced as the DNA strands are being separated. Now, of course, you have to fix the strand, otherwise you're going to create a mutation, right? And so there's ligases come along and seal the nick. Now, the other type of topoisomerase are called type 2 topoisomerases. And this slide represents a model based on topoisomerase 2 in prokaryotes, in bacteria. And that enzyme is called, or class of enzymes are called DNA gyrases. That means bacterial topoisomerase 2. Important to remember. DNA gyrase, bacterial topoisomerase 2. So what's topo 2 do that's different from topo 1? Well, one of the things it does is it actually makes a double-stranded cut in the DNA. Now, if you have a circular genome, you can imagine a double-stranded cut then allows the genome to be twisted, the uncut strand to pass through the cut, and then the cut's fixed. And so basically this will make supercoils. You said, well, wait a minute, you just told us we want to stop supercoils. The trick here is the DNA gyrase makes a supercoil in the opposite direction of the supercoils created by replication. Okay? Replication induces positive supercoils. Topoisomerase 2 introduces negative supercoils. Make sense? So then the physics all works out. Now, how about people? Little more complicated issue in people. Again, people have linear genomes, linear DNA molecules and chromosomes. Chromosomes are not floating around in the cell in solution. The DNA, the chromatin, is attached to stuff in the nucleus. And some of the stuff is indicated here as in green, indicated as chromosome scaffolds. These aren't entirely clear what is involved in this structure, but we know that the loops of DNA, in this example, in, say, an interphase nucleus, are attached to scaffolds. And this is true during replication as well. Now, what does topoisomerase do for you in humans? Well, we think what it does is it, again, introduces double-strand cuts, allows strands to move past each other. The upshot is it allows this structure that's attached to a substrate to be actually to be moved around and to be taken apart and to be replicated and then put back where it was. Okay? So it's a little bit different than in the bacteria. But in both cases, in both humans and bacteria, this has important therapeutic implications, and here's why. And this is an important table. You've got to memorize this table. You've got to memorize the names of these drugs, at least so you can recognize that they are topoisomerase inhibitors. All right, so here's the topoisomerase inhibitor that we met talking about our patient, right? We're going to give the patient doxorubicin to try to get a better therapeutic response. We're going to do that to try to inhibit topoisomerases 2, and in his case, to treat the ALL. Now, doxorubicin also has other chemical effects. If you look it up and read about it, it produces reactive oxygen species, it intercalates into DNA, it interferes with DNA repair enzymes, it does a lot of different chemical things. But we think one of the best correlates with the anti-cancer response is, in fact, inhibition of the topo2 activity. Now, in human cancers, you can also treat with a drug that inhibits topoisomerase 1, different enzyme, but this class of compounds indicated by irinotecan will inhibit topo1 and is useful against these cancers. Here's etoposide. This is a plant alkaloid. Again, topo2 inhibitor, most useful against testicular cancers. And now ciprofloxacin. That's an important one to remember. And this brings up the point that not all topoisomerases are identical. The ones in bacteria are different from humans. And cipro acts on the bacterial topo2, the DNA gyrase, and therefore it's really useful against certain infections. So how much of this do you have to know? You have to know the names of the drugs, and you have to know what their target enzymes are. And I'll tell you a clinical story about such and such happens, and ciprofloxacin was used, and you got this antibacterial effect, and why is that? And the answer is it's a DNA gyrase inhibitor, a topo2 inhibitor. Make sense? Okay. All right. A couple more things before we take a break. So we got the DNA. We're pulling it apart. We're melting it. We're binding it. And now the question is, well, how do you make new DNA from the template strand? And the answer is there's an assemblage of I don't know how many proteins exactly, but dozens of proteins that form the replosome. And this cartoon sort of represents our best understanding, consensus understanding of the replosome in bacteria. The replosome in people is a little different, and I'll show you how and where. But we're going to start with bacteria because we know the most about it, and we're clearest on exactly how this all happens. Okay, so a couple of ideas. There's two strands that are being replicated. There's one called the leading strand, shown here, and one called the lagging strand, shown here. And the way this works is that the DNA polymerase, this sort of olive-looking thing, in bacteria it goes by the name of DNA polymerase 3, an important name to remember, bacterial DNA polymerase. This enzyme recognizes a primer. Let's go all the way back to the beginning here. So early on, this DNA was taken apart, and a primer was synthesized, an RNA primer was synthesized by an enzyme called primase. This polymerase recognized the primer, reads the template, adds the nucleotides, chugs along, and generates a new strand of DNA. All right? Well, that's fine for this strand, but you've got two strands, and how are you going to copy the other one? So on the lagging strand, similar sort of things happen. Primase comes along, makes a primer. That allows the polymerase to land. If you've got nucleotides around, it's going to synthesize a new strand. But you can see in this cartoon, it sort of looks like it's going in the opposite direction. And in terms of the direction on the strand, that's true, it is. And so what happens with this lagging strand is rather than a nice continuous synthesis of DNA, there's a discontinuous synthesis of DNA that leads to what are called Okazaki fragments. Professor Okazaki was a Japanese scientist who discovered this phenomenon. And what happens is the DNA polymerase will chug along, filling in the space until it runs into a previously synthesized primer. It'll let go, fall off, and then as the replication fork advances, frees up more DNA, primase comes in, makes a new primer, polymerase comes along and makes a new piece. You say, okay, that's fine, but now you got DNA with a bunch of holes in it. So what happens next? Well, there's a different set of enzymes that come along and basically fill in the gaps. So the primer is excised by the DNA polymerase 1. That's a different bacterial DNA polymerase. Takes out the primer. The gap is then filled by this polymerase, right? Chugs along, fills in the space. And then the polymerase can't complete the job. It can't go all the way to the end of the template and fill in. Okay? So what it does, it dissociates. And if there's a nick, the nick is sealed by some type of DNA ligase. All right. So this is a little confusing. You say, but wait a minute. How can you have synthesis going in one direction on one strand and the other direction the other strand? What happens to the enzymes? Do they just fall off and float away? No. This is a two-dimensional representation to kind of help you conceptualize that there are two different strands and that there are different sets of enzymes that participate in their synthesis. The reality of how all this works is actually illustrated here. This is called the trombone model of DNA replication. So let me orient you. So here's the DNA that's going to be copied, right? The DNA strands are separated. The leading strand, simple enough, right? It's being copied by good old DNA pol 3, happy as it can be. This whole assembly chugs along, right, and making new DNA, no problem. Now, the trick is to get the lagging strand synthesized. This is kind of cute. The DNA comes through and it actually flips around in 180 degrees so that the enzyme, the polymerase, the clamp loader, and the various accessory proteins are all attached to this assembly, and they're basically all going in the same physical direction at the same time. Pretty cool. Now, if you want to have some fun at the break, go to YouTube and watch how this all works in semi-real time. So what do we remember from all this? What we're going to remember is that we've got leading strand, we've got lagging strand, we've got different proteins on those strands. We need to remember topoisomerase 1, topoisomerase 2, that there are human versions and bacteria versions. DNA polymerases, there are human versions and bacteria versions. I'll show you the human ones after the break. Okay? And that there are drugs that selectively recognize some of those proteins that are involved in replication. Okay? Make sense so far? Good? All right. Let's take 5, and then we'll come back and we'll do the other half of the replication story. So DNA polymerases aren't perfect. If you got a billion bases to copy, mistakes will be made. Okay? A really, really important feature of DNA polymerases is whether or not they have proofreading capability. Can they tell when they've made a mistake? This is going to be really important when eventually when you learn about virology, you learn about HIV, and you learn that some enzymes that make DNA from RNA templates do not have proofreading. And so lots of mistakes get through. And so the virus changes dramatically, rapidly, within days. Now, our genome does not change rapidly within days. It changes a little bit. Every time a cell is replicated, there's an error or 2 or 3 or 4. out of those billions of bases. And that's true also in prokaryotes. The DNA genomes are quite stable, relatively. So why is that? The answer is the polymerases can fix their mistakes. And so what this cartoon is meant is to represent, the little green hand, is the polymerase, okay? And it's reading along the template strand. And it's polymerizing. It reads which base comes next. You know, if it's an A, it goes and gets a T, puts it in there. If it's a G, it goes and gets a C, and so on. And this is the 5' to 3' polymerase activity that we just looked at in some detail. Now, sometimes the wrong base gets added in by mistake. What that does is that creates a structural change in the DNA. Okay, it's the wrong shape. And the enzyme can sense that. And when that happens, conceptually what happens is the enzyme backs up, doesn't physically back up, but it backs up, it cuts out the wrong base, gets the right base, and puts it in there. And that's referred to as a 3' to 5' exonuclease activity and proofreading. So why is it exonuclease? Well, it's at the end of a DNA strand, right? And it's a nuclease because it cuts off the bad base. And it does so in the 3' to 5' direction. That's why that's called that. So what does this mean functionally? Why does this matter? Do a little math here. Everybody had at least a little bit of probability sometime in their background. And so you understand the principle that the probability of a sequence of independent events all occurring is the product of the individual events. Say that again. The product of a sequence of independent events happening is the product of the individual probabilities. Okay, so what are we talking about? Well, here's one event, the polymerization event, error about 1 in 10 to the 5th. Yeah, that's not too bad, but still you got to make 10 to the 9th reactions. Okay? So there's going to be a couple of errors in the human genome. The proofreading, 1 in 100. You go, oh, that's not exactly high fidelity, but it's good enough. And a third event, which we haven't met yet, called strand-directed mismatch repair, also an error rate of 1 in 100. So what that's about real quickly is as this machinery is going along making the DNA, the new DNA, if the two strands are mismatched, if they don't have exactly the perfect complementarity, then it creates kind of a bulge, and there's a whole family of proteins that zoom in and fix it. And we'll talk about those tomorrow. All right, so the combination of all these three events resulting in one mistake getting through is 10 to the 9th. Okay? So 5 plus 2 plus 2 is 9. Got it? So the fact that the polymerase can fix things, the fact that there's a fairly low error rate to begin with, and the fact that there are other DNA repair enzymes that also fix things means there's a very low incidence of errors, but not zero. Okay, so mutations do get through if they confer an advantage. If they go on, they get transmitted. If they create disease, depending on the type of disease, they may stop at that generation, or they may become an inherited trait, sickle cell disease, okay, thalassemias, and so on. Does it make sense? All right. Now, let's go from prokaryotes and talk specifically for a couple slides about human DNA synthesis. When does it happen? Well, it happens in the S phase of the cell cycle. So I'll pop up the cell cycle to remind you that there are different phases. Everybody probably remembers mitosis we just kind of talked about, right? Segregation of the chromosomes. DNA synthesis in mammalian cells occurs in S phase. DNA synthesis in bacteria can occur a lot of the time. It's not organized into a nucleus, and it's not necessarily in the specific phases of a cell's life cycle. But in humans, it's in the S phase. So this is going to matter for a couple of reasons. One important one is that there are drugs used to treat hyperproliferative diseases and cancers that work best in S phase. And you think, why might that be? Well, because they inhibit DNA synthesis, okay? And that occurs in the S phase. Now, in humans, the DNA synthesis is catalyzed by functionally homologous enzymes to the bacterial pole 3 and DNA pole 1, but they have different names. And there's more of them. And I don't know if that means bacteria are smarter than us. They don't have to make as many proteins to do the job, but that's how it works. And they do slightly different things, okay? So, some examples that you need to remember. DNA polymerase alpha in humans has the primase activity. Remember, that's what was required to start the DNA synthesis. Okay? The pole epsilon, arguably, is probably the most important one of this whole group, and it synthesizes the leading strand, and it's also involved in DNA repair. Important. Okay. Mitochondria have their own DNA, and they have a polymerase that catalyzes that reaction. Okay. And here's a pole B, which is a DNA repair enzyme, and importantly, no proofreading in these first two. Remember, the point I made was that the very important feature of these DNA polymerases is whether or not they have proofreading. So you need to be familiar with this table, recognize if I'm talking in a question I'm talking about, if one of the choices is between DNA polymerase 3 versus DNA polymerase epsilon, understand that pole 3 is a bacterial enzyme and epsilon is a human or mammalian enzyme. All right. And understand that because polymerases are slightly different proteins, different enzymes, they are somewhat differently sensitive to drugs, to inhibition by drugs. So we're going to talk about a couple of examples here to illustrate that. So what you see in front of you are nucleoside analogs that inhibit DNA polymerases, and these are two compounds that are used in medicine. The first, zidoviadine, or azidothiamidine, looks a lot like good old regular thymidine, right, which is a pyrimidine. Everybody remembers that. Here's a pyrimidine ring. Okay, and this is the deoxyribose. But now instead of a 3' hydroxyl, it's got this azido group. So that should ring a bell. 3' hydroxyl. Why is that important? Remember we talked about primase and the RNA primer? Well, one of the key things in that primase and the primer that's absolutely required for DNA synthesis replication is a 3' hydroxyl, because the polymerase needs to see a 3' hydroxyl coming off of the deoxyribose. That's an important physical determinant of allowing that polymerase to work. So now we've got a drug that doesn't have a hydroxyl, so could be something up there. All right, the other example is dideoxyinosine, important for a couple of reasons. It's used in DNA sequencing reactions, but medically, more importantly, it is a drug that's used to treat HIV infections. And it is a polymerase inhibitor. Now look at this. It's missing both hydroxyls, hence the name, dideoxy. And it looks a lot like good old deoxyadenosine. So the theme here is that molecules that kind of look like native nucleosides and nucleotides are sometimes pretty good polymerase inhibitors. Now the third example that I want to talk about using a clinical case is a drug called acyclovir. Now the story here, following up on the case we started with, is your patient PL, diagnosed with the Philadelphia positive ALL, and he's now being admitted for a bone marrow transplant. Now this is essentially a curative treatment for this disease. The deal with bone marrow transplant is the physician has to be sure that the patient doesn't have an active infection at the time of the transplant. Right? That's one of the problems that they have to sort out. So after lab tests, this person was positive for antibodies against the herpes simplex 1, HSV1 virus. And this is a virus that causes some stomatitis, can cause basically cold sores around the mouth. And that's illustrated up here. All right? And his history says that 4 months ago he had this infection. So this is a problem for the transplant. But the issue here is, well, what could be done? And the answer is this patient can be treated with a drug called acyclovir. So here's some medical jargonology for you. IV means intravenous. Here's the dose, mass per unit, mass of drug per unit body weight, milligrams per kilogram, given every 8 hours for 7 days. All right? And that's an example of how this drug would be given. So what is this stuff? What does it do? Well, here's the structure of acyclovir. So purine or pyrimidine? What's it look like? Purine or pyrimidine? Purine, right? Double ring structure. But, got a sugar? No sugar. So deoxyguanosine, the native purine that it most resembles, right? Here's deoxyribose. So this is a nucleoside, right? No phosphates here. If it's phosphorylated, it's a nucleotide. In order for this drug to work, in fact, it has to be phosphorylated to a nucleotide. And here's the trick, which makes this drug so cool. This drug is metabolized by an enzyme coded by the viral genome, thymidine kinase. You say, well, wait a minute, it's not thymidine. Yeah, but it's recognized by viral thymidine kinase, and it's phosphorylated. So only cells, this is the trick, only cells that are infected with the virus express the viral thymidine kinase. Right? You're not a virus. Okay? So the drug in the cells that are infected gets metabolized to this stuff, acyclo-GTP. Right? Here's the phosphate, 1, 2, 3. Guanosine, acycloguanosine triphosphate. So it sort of looks like deoxy-GTP. So if it sort of looks like deoxy-GTP, what happens during DNA replication? Well, the polymerase will recognize acyclo-GTP, says, looks like I need a deoxy-GTP. Close enough. And then incorporates it into the strand, right? So here's the template strand, here's the growing strand. Now, there's no sugar. So what happens at this point? We got no 3' hydroxyl. So the polymerase stops. Key point, polymerase stops because there's no 3' hydroxyl. You get what's called chain termination. And that's the basic molecular biochemical mechanism of a variety of DNA polymerase inhibitors. It causes chain termination because they look enough like the native base, they get incorporated in the chain, but they're not right. Sometimes the polymerase can continue on with other structures, not this one, but other types of drugs, and you have the wrong base in the DNA. So what happens then during gene expression? You don't make the right messenger RNA, you don't make the right protein. So these drugs can actually have multiple biochemical effects. But the one I want you to remember for this one is chain termination by an analog that is missing a 3' hydroxyl. Okay, make sense? All right. So you guys are all pharmacologists now. Pretty cool, huh? Okay, so where do we go next? All right, so we've replicated DNA and then therapeutically have inhibited it in the virus. But completing the replication of a cell and going through the cell cycle and successfully making a daughter cell takes more than just copying the DNA. The cell has to copy the structure of the chromatin. Okay? So how does that happen? It's a little complicated. So remember that the DNA is not floating around free like a Watson-Crick vDNA model. It's actually bound up with histones. The little yellow dots are meant to represent the histone octamer, 8 subunits, right? 200 bases wrapped around there. That's called the nucleosome. Some of the DNA, euchromatin, is associated with these histones, and not a whole lot else. Some of the DNA, the heterochromatin, is associated with a variety of binding proteins shown here, just sort of generically described as heterochromatin proteins. There's a bunch of things. The point is that the chromosomes need to be copied so that this structure is replicated into the daughter cell. And how does that happen? Well, it happens by a fairly complex biochemical machinery, the details of which we're not going to delve into at all. After the replication of one of these strands, the pattern of binding proteins is retained to the degree that it can be copied in the daughter cells. Okay? So that's the basic concept. But what about this histone modification? What does that mean? And I need to stop here and try to use this little pencil thing. So bear with me. All right. So, in the histone structure, remember these yellow dots are octamers. And so each of the mers is a particular histone. And that histone is basically a globular protein, and it has an N-terminal tail, and it has a bit of a C-terminal tail. It's essentially a globular structure. Now, the key to this modification story is that in the N-terminal tail, there are amino acid residues in there that can be acetylated, methylated, or phosphorylated by enzymes, right? So histone acetyltransferase puts the acetyl group on the histone residue. Histone deacetylase, HDACs, take it off. And similarly, there are methyltransferases to put the methyl on, demethylases to take it off, protein kinases to put a phosphate on, protein phosphatases to take it off. You say, okay, well, that's swell. Why are we doing this? This creates what's called this pattern of covalent modifications in both the N, primarily the N-terminal, but also some in the C-terminal, creates what's called the histone code. So you know about the DNA code, right? Codons, coding for genes. On top of that, there's what's called an epigenetic code, comprised of the covalent pattern of covalent modifications of histones, the histone code. That code also has to be faithfully replicated in order for the daughter cells to look like the parent cell. Why is that? That's because these modifications control the pattern of gene expression. So part of the reason that your skin cells look like your skin cells, your heart muscle cells look like your heart muscle cells, your kidney cells look like kidney cells, is because of the sets of genes that are turned on and off, and those are controlled in part, importantly, by this histone code. That make sense? Yes, sir.
You're saying that heterochromatin proteins are replicated after the round replication, proteins are replicated and expressed to go back to the original place?
Yeah, all except for the after. It's actually done at the same time. So what you can imagine is in the heterochromatin, the replication machinery is kind of coming along, right? And it gets to the heterochromatin, and some of that dissociates by mechanisms we don't clearly understand. And that pattern is copied or remembered by this overall machinery. And then the heterochromatin proteins, which have been synthesized and are in the cell already, they are put back as the DNA strands are matured and synthesized. Okay, so it's not stepwise, it's actually simultaneous. But basically, yeah, what you said is true. Okay? And that's for the heterochromatin binding proteins. But what about the histone code? Well, the histone code is then rewritten by a complex called the reader-writer complex. It's not a terribly important detail, but that's the name of it. And again, complex set of proteins that essentially read the code from one strand and replicate it and reproduce it so that these strands look the same as they did in the first place. Now, this is all terribly complex, and we're not going to go into the details. The take-home is histones can be modified primarily on their N-terminal tail by covalent modifications. What are the names of them? Acetylation, methylation, phosphorylation. What do they actually do? It varies, but generally, acetylation activates gene expression, methylation represses. Okay? And to get the right set of proteins in the daughter cell, you've got to copy the histone modifications as well as the heterochromatin binding proteins. Okay? Make sense? Okay. So, once the DNA is replicated and the chromatin structure is reestablished, it's all transferred into the daughter cells. Life's good. They go along. They do what they're supposed to do. But cells and people get old, and they die. And there's a finite lifespan. Why that is not completely understood scientifically, but in the recent past, a really important clue was provided by understanding what this structure called the telomere does. The telomere is a piece of heterochromatin at the very, very, very end of the chromosome. Okay? We're going to talk a little bit about the details of what that is. The telomere, being the last bit of DNA, actually has a recognizable and repeated pattern called the telomere repeat. So if you look at this strand, you can see TT, GG, GG. TT, GG, GG. That's the telomere repeat, one of those. And in some telomeres, there are thousands of them. Some there are hundreds. In some chromosomes, there are very few. Okay? So, why does this matter for lifespan? Well, the concept that emerged, again, in the 90s was that this was part of the biological clock that dictated how long a cell lived and therefore how long an organism lived. So, in general, but not always, longer telomeres, longer lifespan. As the telomere burned off, for somehow, we don't know exactly how, this often triggers senescence, meaning the cell stops growing, and eventually, if the cell remains senescent long enough, it will die. Okay? So how are the telomeres maintained? And why would they need to be maintained? Well, if you think about it, back to our patient PL who has this acute lymphoblastic leukemia, before, or other people who don't have this leukemia, they have lymphoblastic cells, right, that you need to make lymphocytes. And these lymphoblasts in the bone marrow tend to have stemness or pluripotency. They continue to grow for a long, long time, your whole lifespan, 80-some-odd years, 90 years. And in those types of cells, those stem cells, and in your patient's cancer cells, what happens is they have very high activities, or they have significant activities of an enzyme, telomerase reverse transcriptase, H-TERT. And that's the little green boat that you see here. So what does H-TERT do? It's a really weird enzyme. It actually carries along with it a little piece of RNA. And it's a reverse transcriptase. What does that mean? That means it's an enzyme that uses RNA as a template to make DNA. Okay? DNA polymerase uses DNA as a template to make DNA. Reverse transcriptases use RNA. That's a very important distinction. All right? So, what this enzyme does is it recognizes the telomeric repeat. The RNA template binds to it. The enzyme synthesizes DNA sequence here in red. See that? So it makes more telomeric repeats. TT, GG, GG, TT, GG, GG, and so on and so forth. Then what happens is DNA polymerase kind of backfills the complementary strand, and you have a telomere that's maintained. Okay? And presumably lifespan is maintained. Now you think, well, is this a good thing or a bad thing? Well, if you're a bone marrow progenitor cell, it's a real good thing. If you're a cancer cell for the patient, that's a bad thing. So HeLa cells have telomerase activity, right? And they've existed in culture for more than 50 years. We invented in the late 90s immortalized human airway smooth muscle cells that people use to study signaling in the lung. And they've grown since 1990s because I put telomerase, reverse transcriptase, into the cell with a virus. Henrietta Lacks had a virus that transformed her genome, and her cells remain telomerase positive, and that's part of the reason they've survived as long as they have. Okay. All right. The other, the structural importance of telomeres and telomerase is this thing called the T-loop. So this is the end of the chromosome, all right? And so somewhere in here are the telomeric repeats, and what happens is these telomeric repeats will invade somewhere upstream. They'll invade the DNA strand, and they'll form something called a D-loop, which we'll learn about in a minute when we talk about repair, or recombination, rather. So this makes a loop at the end rather than having a free end of the DNA. So why is that a good thing in terms of DNA stability, genomic stability? If you've got free ends of DNA sticking out, there's a whole class of enzymes called nucleases, right? Exonucleases. They're going to come along and they're going to chew up the DNA. So this protects the DNA from nuclease degradation. That's probably the major function of the telomere. And again, when the telomere is long enough, that protection lasts a long time, the genome is stable, the cells last, they don't senesce, and so forth. Okay? All right. So we're going to take another break here before we come back and do genetic recombination. But everybody clear on what we need to know on all this stuff? Lots of details. Some new ideas, I think, for some of you, right? Telomeres, breeder-writer complex, histone modifications. Okay? Let me ask you, is the background reading, is that helpful at all for this? Too much, too little? It's okay? I know some of you have your own books from molecular biology. It probably covers a lot of the same stuff, right? But the twist, the difference is when I ask you test questions, they're going to look more like the first slide I put up in the lecture. Patient X, Philadelphia positive, has extended telomeres. Why could that be? And you should look for an answer, he's telomerase positive, something like that. You see the difference? As opposed to, what does telomerase reverse transcriptase do? It's sort of assumed that you know that, and you can use that to solve a problem. See the difference? Okay? All right, let's take 5 or 6 minutes, take a break, stretch. How you doing?
Hey guys, real quick before you go.